So databases. So in the previous lecture, we discussed the strategies used to sequence genomes. We also discussed how the human genome project required innovations in the sequencing techniques, in the computer hardware and software. And we also mentioned that the sequences that are obtained by sequencing the genomes of human individuals or other organisms are stored in databases, and the data in these databases could be used for example, for identifying or studying genes. So one way of identifying a gene or studying a gene is first by obtaining a sequence for that gene and then comparing the sequence of that gene to sequences already stored and already stored in the database, and already these sequences are known for specific genes. So you have, for example, you have a gene sequence and you want to know the gene function. So you want to learn something about the gene function or have some information about the gene function, okay? So what you will do is that you take this sequence, okay, and compare it. So you do something called blast search. So blast search, this is like a Google search, but instead of searching for words, you are searching for a gene sequence. So you put the gene sequence, and a software will compare the gene sequence, your gene sequence, with gene sequences already stored in the database. Okay, so this will compare your gene sequence with the millions of sequences stored in the databases. So sometimes you will get a match or a result, and from that result you might identify what type of protein encoded by your gene, for example, okay, and maybe have some information about the gene function. And this process is called gene annotation. So gene annotation is basically a process of identifying genes and determining their functions, okay? So before the Human Genome Project, before the development of genomics and bioinformatics, gene annotation was carried out using experiments in the lab. And it took maybe months or sometimes years to annotate a gene, to identify it and determine its function. But now you can annotate genes by comparing them to sequences in the databases in a matter of minutes or maybe seconds, okay? But still, even if the blast search gives you an idea about the gene name or the gene function, sometimes you have to go and do some experiments in the lab to make sure that the function suggested by the blast search is actually valid, or to maybe you have to go and do some experiments to confirm the gene function or the gene identity. Not only the blast search could be done for gene sequences, but also could be done for protein sequences. So both, if you have a protein sequence or amino acid sequence or nucleotide sequence, it's the same. So before the Human Genome Project, for example, some genes were already known and their functions were known, but some other genes were unknown and their functions were unknown. So what happened is that after the Human Genome Project, scientists identified new genes and they compared the sequences of these new genes with the sequences of other species. Okay, so the blast search will compare your gene sequence with sequences in the database. If you have a human gene sequence with unknown function, you can compare this human genome sequence with all sequences from different organisms. Okay, so sometimes the blast search will give you basically a result that shows that this human gene is similar, maybe, to a mouse gene. And this mouse gene, basically, already the scientists determined its function with a known function. So for example, let's say that you have 99% similarity between your human gene sequence and a mouse gene sequence. But the mouse gene sequence encodes a protein with a known function, which means the human gene or your human gene might have the same function. Okay, so we can compare gene sequences, unknown gene sequences, or unknown gene or gene sequences of unknown genes with all the gene sequences that are found in the databases from all sorts of life, or all from different organisms, or hundreds or maybe thousands of organisms. Okay, because you have to understand, many of the genes we have inside our cells, they have other organisms, they have similar genes encoding proteins with similar functions. Of course, gene annotation using computer softwares and databases could be used as the first step to identify or annotate the gene, but you have to do some functional studies or experiments in the lab to confirm the function or the identity of the gene in question. Okay, so one type of functional study is to do something called knockout. So basically, and we discussed this in the previous chapter, one strategy to basically to identify the function of the protein is to knock out. So knock out the gene. Okay, knock out the gene means you completely delete the gene delete or disrupt the gene, so that the gene is no longer functional, and this might lead to a mutant phenotype that might give you an idea about the function of the protein or what type of processes this protein might be involved in inside the cell. Okay, so after you do gene annotation using searching the databases and computer softwares, you could go and do functional studies. One type of functional study is, or one strategy of doing functional studies is to knock out the gene function, delete the gene completely from the genome or disrupt it or destroy it so that mutant phenotype will be generated. And studying the mutant phenotype might give you an idea about the function of the gene. Okay? So the mutant phenotype might give you an idea about the function. Or confirms, basically, the annotation carried out by the blast search. Okay. CRISPR-Cas9 can also be used for such knockout experiments. There are also some areas of research are focused on studying the regulation of expression of all genes, maybe during development or during basically adulthood. So in 2003, for example, following the Human Genome Project, an initiative by a group of scientists was launched. And this initiative called the ENCODE project. So the ENCODE project began in 2003 and ended 2012. And the aim of this project is to determine all or study and determine all the DNA elements required for the expression of genes. Okay. Here we're talking about promoters. So they wanted to identify all the promoters of genes, enhancers, and silencers, and many other types of DNA elements. Okay? So basically, they wanted to learn how these DNA elements direct the expression of genes. Now, also in this project, they wanted to understand how histone and DNA modifications might affect the chromatin structure and how these modifications might have an effect on the gene expression. And after this project, the scientists found out that 75% of the genome is transcribed at some point. Okay, and most of these, of this will result in non-coding RNA. So we know that 1 to 2% of the genome in humans encode proteins. So scientists, after discovering that coding sequences or genes represent only 1 to 2% of the genome, they thought that this is just the only DNA that is transcribed. But after the ENCODE project, scientists found out that this is not the case. Actually, this number is actually 70% of the genome is transcribed, mainly to non-coding RNA. Okay, at some point, okay, different regions are transcribed at different points in the life of the organism. Okay. There was also another genome following the Human Genome Project, which is to provide a map for the human epigenome. The human epigenome basically is all the modifications that happen to the chromatin inside our cells, so to the histones and to the DNA, and where these and how these modifications affect the gene expression. Okay? Another branch of science that was also developed following innovations in computer technology and sequencing technologies and in biotechnology in general is the systems biology. So now we have something just like we have a genome. Okay. And we have genomics. So genomics is the study of the genome or the genes in the genome and how these genes are interacting with each other. Now we have something called the proteome, which represents all the proteins expressed or found inside a cell, or maybe a group of cells, or maybe a tissue. And proteomics is the studying all these proteins and their properties and their interactions. Okay. Now, systems biology is basically the study of whole biological system. So it's the study of a whole biological system. Nizam biology, for example, studying the whole cell. So a cell could be considered a whole biological system. In system biology, we try to understand how a whole biological system, like a cell, behaves by studying the interactions between the different parts of the system. So we could study a whole biological system like a cell by studying the interaction, تفاعل, between the different parts of the cell. Different parts here could be the genes, could be the proteins, could be something else. Okay. Now, the amount of data generated by studying a whole biological system is very huge. Okay, or the amount of data is very huge and requires softwares to analyze this data. You have to understand, if you want to study a very complicated system, نظام معقد, like a cell, you need to study first the different parts, and then you need to study how these different parts are interacting with each other and how they affect each other positively or negatively. So you can think of one strategy of systems biology is to study how genes interact with each other or how proteins interact with each other and how these interactions will allow the cell to carry out its intended functions. Okay? And you have to understand that the systems that we are talking about, biological systems, they are dynamic, متغير. They're not static. They're not static or ثابت. They always change. So we need to find a way to see how this system might change upon a specific situation or a specific stimulus or a specific response. Okay, so for example, scientists applied the system biology approach to an organism called Saccharomyces cerevisiae. Saccharomyces cerevisiae is a type of yeast هي الخميرة. So in this study, they wanted to understand the interaction between genes. So what they did is that they deleted or disabled or knocked out pairs of genes, and they basically observed how deleting both genes might have an effect on this type of organism, and what type of mutant phenotypes might be observed or might be obtained. So then a computer software analyzed, so we have something called double mutants. Double mutant means these are mutants, they have 2 deleted genes, double 2. Then, of course, there will be thousands of genes in the yeast, and there will be, thousands of different options of deleting 2 genes at a time, and observing how the deletion of each 2 genes will affect basically the phenotypes of this yeast. So the next step was to use computer softwares to analyze the different pairs of genes deleted and the phenotypes, the mutant phenotypes generated. And this was the approach to come up with a map, okay, that will basically allow scientists to draw conclusions about the interactions between the genes in this type of organism. Okay? So a computer software produced a network-like functional map. Functional map means this is a map that will show which genes interact with which genes. Okay, and this is the result of the study. So here, as you can see, this is the map. This is the functional map we are talking about. And in this map, as you can see, each dot represents a gene, and each line represents an interaction between these genes. Okay, so these genes, they are color coded. So each, let me zoom in. Okay, so as you can see here, each dot, whether it's white or colored, represents a gene, and these colored genes, they are involved in similar functions. So here we have the orange dots represent genes that are important for nuclear cytoplasmic transport. The green genes, the dark green genes, are important for mitosis. The light green genes, they are important for DNA replication. And the lines between them represent interaction between these genes. So these genes affect each other, or they work together in the same function. So if we zoom in to this region, here we have genes that are colored blue, and these are genes important for metabolism and the synthesis of amino acids. And as you can see, if we zoom in, here we have a number of genes. These genes, for example, these maybe 10 genes are important for the synthesis of glutamic acid. These genes are important for synthesis of the amino acid serine. And here, these genes, each one of these genes, as you can see, there are some lines extending and connecting to other genes, which means that these genes are interacting with each other. These lines, these white lines, they represent interactions between these genes. So as you can see, there's an extensive network of interactions, very complex interactions between these few numbers of genes. Look, this is a whole, this picture or this map, this functional map shows all the genes in yeast and the interactions between them. Okay? Of course, scientists will not be able to draw this map or to draw conclusion from this map without the help of very complicated computer software or very powerful computer software. So what benefits we might get from systems biology, from studying the different parts and their interaction within a very complicated whole biological system? It turns out that we could use system biologies to study cancer and determine which genes are involved in cancer and how the gene expression in cancer cells, okay, differs from the gene expression in normal cells. Okay. Of course, this was done through a project called the Cancer Genome Atlas, which in this genome, scientists, they use system biology to try to look for the major differences between cancer cells and normal cells. And this project, they were able to determine the most common mutations responsible for the transformation of a normal cell into a cancer cell. They also looked for the differential gene expression between cancer cells and normal cells. Okay. And this was done with also using a technique called microarray. We already discussed microarray in the previous chapter. Just as a reminder, microarray is a technique used to study the gene expression, the expression of all genes inside the cell, for example, or a tissue or a sample. So microarray was used in this project to study the differential gene expression between or the differences in gene expression between cancer and normal cells. The next concept in this chapter is about the properties of genomes from different organisms. So genomes from different organisms, they have different sizes, they have different number of genes, and the gene density, cathephy. And now we could study the genomes of many organisms because, again, scientists have successfully sequenced the genomes of thousands of organisms, different organisms. And this is made possible because of innovations in the DNA sequencing technologies and computer softwares, computer hardware. So let's talk about first the genome size. The genome size varies from, a couple of thousands of nucleotides to billions of nucleotides. So for example, most bacteria, they have a genome size that ranges between 1 to 6 million bases. Genomes of archaea, they have similar size range, but generally speaking, eukaryotic genomes, they are much bigger and they are larger than prokaryotic or bacterial and archaic genomes. Okay? So for example, many plant and animals, they have genomes greater than 100 million bases. Humans, for example, the haploid human genome consists of 3 billion or 3,000 million bases. Okay. This table, for example, lists some of prokaryotic and eukaryotic organisms and their haploid genome size. Okay. So as you can see, some types of plants, they have genome sizes even bigger than the genome of the humans. Okay, so here we have the scientific name, and in between brackets, بين الأقواس, we have the common. So the scientific name for humans is Homo sapiens. Also, in this table, as you can see, we have the number of genes within the genome. So we have approximately more or less 21,000 genes. Okay, and if you divide this number over this number, you will get the gene density. Okay. Okay, so again, here in this table, we have the haploid genome size in megabase. So this number you have to multiply it by 10 to the power 6. So for example, the human haploid genome consists of 3,000 megabase. So 3,000 megabase is equal to 3 billion bases, okay, or 310 to the power 9 bases. Okay, so megabase equals 10 to the power 6 bases. And in general, eukaryotes, they have larger genomes, but a large genome does not necessarily mean more genes. So, for example, if you compare the genome of a nematode, C. elegans, with the genome of Drosophila, so the genome of the Drosophila is larger. So if you compare this gene, this organism, Drosophila is the fruit fly, with the genome of this organism. So as you can see, the genome of the Drosophila is larger, but it contains less number of genes. So here we have 165 megabase, 100 megabase, but here we have lower number of genes. And Drosophila, it's a more complex organism than the C. elegans. So why do you think a more complex, how can a more complex organism like Drosophila can basically have less number of genes compared to a less complex organism? The answer is that these 14 genes, 14,000 genes, they can produce, okay, more proteins than these 20,000 genes. Do you know how? So although Drosophila has 14,000 genes, they can produce more genes, more proteins from these genes compared to the compared to the genes in this similar organism. Do you know how? How a Drosophila can produce more proteins from a lower number of genes? So this is because of alternative splicing. So remember, alternative splicing allows the generation of more than one polypeptide from a single gene. So for humans, before the Human Genome Project for example, before sequencing the human genome, scientists estimated that we will have 50 to 100 thousands of protein encoding genes. So this is before. Why do you think scientists, they estimated that we might have 50 to 1,000 to 100,000 of protein encoding genes? From where did they get this number? Do you know why? Or from where did they get this number? So on average, when they analyzed the number of different proteins in a typical human cell, they found that it contains 50 to 100,000 genes. Okay, on average. But after the Human Genome Project, so Human Genome Project, we know that protein encoding genes are almost 1/5 this number. So how can this lower number of genes encodes this large number of polypeptides? Again, because of alternative splicing. Okay. So again, before the Human Genome Project, scientists estimated that human cells might have between 50 to 1,000 to 100,000 genes encoding proteins based on the number of proteins found in the human cells. But after the Human Genome Project, scientists were shocked to discover that the number of genes is even less than this, and alternative splicing explains how this lower number of genes can produce this large number of proteins. Okay, same thing. By analogy, this is why Drosophila can produce more proteins than C. elegans, although they have lower number or less genes. Okay. We have something called the gene density, which is basically the number of genes in a given length of gene. So here we have the gene density, the number of genes for every 1 million bases. Okay. So for humans, if you divide, so this is the number of this is the number of bases in megabase. So we have 3,000 megabase. If you divide this number, 21, over this number, you will get approximately 7 genes every 10 to the power 6 bases. This is, or every 1 megabase. Okay, this is that. So as you can see, in general, bacteria and archaea, they have a higher gene density. So the gene density in mammals, like humans in general, is less compared to the gene density in other eukaryotes or even in prokaryotes. So prokaryotes in general, they have higher gene density. There is no introns in prokaryotes, and genes are parts of operons, for example. They are very close to each other, and there is no large non-coding DNA regions between genes. But in large animals like humans and mammals, we have most genes, or a large number of genes, are mainly made of intron sequences. So intron sequences dominate, for example, in a human gene, in a typical human gene, or in most human genes. Also, we have large non-coding sequences within, let's say, between genes in humans, for example, or in higher organisms. So, for example, in humans, on average, بالمعدل, human gene consists of 27,000 base pair. Okay, of these, or a human gene, a bacterial gene, on average, it's a 100 base pair. So human genes, they are larger, they are 27 times larger than a typical bacterial gene. Okay. Here, because most of this gene, okay, will have nucleotides in introns, not exons. Also, there is a large number of bases or large pieces of non-coding DNA exist between the human genes within the genome. In bacteria, the genes are very close to each other. Okay. So in the next concept, we're going to learn about the structure of the human genome. So far in this course, or in this chapter, we have been focusing on genes that code for proteins. But these genes that code for proteins, they only represent 1 to 2% of the genome. So only 1 to 2% of the genome codes for proteins. Okay. So what about the rest of the genome? What about the 99, almost 98 to 99% of the genome? So 99 or 98.5% of the genome, okay, so the rest of the genome represent 98.5%, let's say, more or less, does not code for proteins or ribosomal RNAs or tRNAs. So here for proteins, rRNA, and tRNA. So it turns out that regulatory sequences, here we're talking about promoters, enhancers, silencers, and many other sequences that are important for the expression of genes. So regulatory sequences, they count for 5% of the genome, and introns, they account for 20% of the genome. Okay. So most of the DNA is non-coding DNA. And the non-coding DNA that is found between genes includes pseudogenes. These are genes that used to have function and they accumulated mutations and now they are non-functional. Also, we have repetitive DNA. So we have certain sequences that are repeated over and over in the same location of the chromosome or in the genome or in multiple locations in the genome. So we have this pie chart that shows the types of sequences that are found in the human genome. So as you can see, exons, they only represent around 1.5%. The regulatory sequences of genes like enhancers, promoters, silencers, and many control elements, they represent 5%. Introns, they represent 20%. Now, in general, we have also repetitive sequences. Now, these repetitive sequences, 70% of repetitive sequences sorry, 4% of the genome consists of repetitive DNA that consists of transposable element, transposon, and their sequences. And we will talk about these transposon, inshallah, this chapter, maybe next week. And then we have DNA repetitive sequences that are not related to transposable elements or transposon. We have certain large duplications, which represent 5 to 6%. We have simple repetitive sequences, 3%. And then we have the transposon repetitive elements, and there are two major types: L1 sequences and allo elements. So L1 sequences, or they are actually sometimes called line 1. Line is stands for long interspersed nuclear elements. So these are transposable elements. They represent almost 70% of the genome. They are responsible for many of the insertions, deletions, rearrangements that might happen inside the genome. Okay, and typically within the genome, most of these sequences or transposons, they are inactive. Okay, so a typical L1 element consists of approximately 6,000 base pair, and these 6,000 base pair basically might have, they might have non-coding sequences, and sometimes they might have some genes that might be expressed, and these are repeated or found in multiple copies throughout the gene. Some scientists found out that there is these sequences, they might play a role in certain types of cancers. Okay. The L1, so each L1 sequence is around 6,000 base pair, but there will be thousands of allo sequences scattered and repeated throughout the genome on almost all chromosomes they are found. We have also allo-1 elements, which are repetitive DNA sequences. Also, they represent 10% of the genome. These elements, they are repeated thousands of times within the genome, and each allo element consists of around 300 base pairs. So sometimes they are called sign. Sign for this is long interspread nuclear elements. This is short interspersed nuclear elements. Scientists estimate that there is 1 million of these elements within the human genome, scattered and found repeated in throughout our genomes on all chromosomes. Okay. And then we have this unique non-coding DNA region, which represents 15%. Okay. And these are DNA regions that are found in unique, and they are basically highly conserved sequences that are found in many organisms, but they do not code proteins. Okay, so you might find, let's say, similar genes with similar sequences in different organisms, like in humans, in mice, and rats. But these, because they are genes encoding similar proteins carrying out several functions. But these are non-coding DNA that is still very similar or identical in different organisms, and still scientists trying to understand why these are identical nucleotide sequences are found in many organisms and what they actually do. So now let's talk about transposable elements and their related sequences. Again, they represent 44% of our genome. So we have to learn about this. So 75% of the human repetitive DNA is made of these transposable elements. So this is 70% of the human repetitive DNA, not the human genome. Okay. So the human genome, almost we have 75%. Sorry, you have 1.5%, 5, 20, and 15. Okay, what is left is almost let's say 40, maybe 48.5%. Actually 58% or 60, around 60% consists of repetitive DNA. Of this, so this is repetitive DNA sequences that are repeated over and over in the genome. Okay, and 75% of these are, and 25% are repetitive DNA, so transposons, and their related sequences. So 75% of the repetitive DNA, which represent 44% of the whole genome, are transposable elements and their related sequences. The rest, the other 25% of the repetitive DNA, they are repetitive DNA unrelated to transposable. So when I say 75% of the repetitive DNA, not the whole gene. So these transposable elements, sometimes they are called jumping DNA or jumping genes. Jumping means بنط in مكان from one, so they can move from one place within the genome into another. So the first scientific evidence for these mobile elements, متحركة, came from studies done on maize or Indian corn, by a female scientist called Barbara McClintock. So Barbara McClintock was studying the change or the different, how the color of corn, Indian corn, keeps changing, and why it changes, and why the same corn ear will have multiple colors, as you can see here. And Barbara was trying to explain why different, or why there is variation in the within the color of the kernels on a corn cup. So this is a corn, okay? So a corn cup will have more than one color, for example. Some are, as you can see, yellow, some are maybe blue, some are more, they might be purple, and something like this. So as you can see. Of course, Barbara, she was studying corn in 1940s to 1950s. And she identified, or she provided explanation for the change in the color of corn kernels from one generation to the next. So she kept tracking the change in the color of corn kernels from one generation to the next. And she suggested that there are some DNA elements or mobile genetic elements. Nowadays, we know that they are called transposable elements. They can move from one location within the genome into the genes important for determining the kernel color, so that these genes will be disrupted. Okay, so the idea here is that within the genome of the corn, there are multiple genes that are important for determining the color, the kernel color of the corn cup. So these are the kernels. These are the kernels. And so these are, let's say, genes important for kernel color. Kernel color in core. And imagine if this is the genome of corn. So corn genome. Of course, it's not circular, but bear with me. And there is a transposable element. So this is transposon or transposable element. Trans mean متحرك. Poson or transposable element, it changes its position. That's why it's called transposon. It can move from here, for example, and maybe put itself in some of these genes, disrupting these genes, okay, and causing a change in the color of the corn kernel from one generation to the next. Of course, in the 1940s and 1950s, when Barbara, this is an image of Barbara, when she suggested that there are some DNA elements or DNA sequences that can jump from one place in the genome into another, other scientists, they thought that she was crazy, actually. And they did not believe her. But later on, more studies by other scientists confirmed the ideas suggested or the hypotheses suggested by Barbara. And that's why eventually in the 1983, when she was 81, okay, she was awarded the Nobel Prize for her discovery of transposable elements. Okay, so this is a typical story where a scientist discovers something and other people or other scientists, they think he or she crazy, and later on they find that he or she was actually correct in the first place. Okay. So it turned out that these transposable elements are of two types. We have transposons and retrotransposons. Transposons, they move from one location within the genome through a DNA intermediate, and they require an enzyme called transposase. A retrotransposon moves by a means of an RNA intermediate. Okay. So this figure for example, shows how a transposon moves within the genome. Okay, so first, a transposon will make a copy of itself. Okay, and this copy will be mobile, which means it can move, and the new copy is inserted elsewhere in the genome. Okay, so now we have two copies of the transposon, and these two copies, they have identical or very similar DNA sequences. So this is called copy and paste mechanism of transposition. Transposition is basically the movement or the change in the position of the DNA. So copy and then paste. The transposon is copied, and this copy is pasted elsewhere in the genome. For retrotransposons, also a retrotransposon is basically transcribed into an RNA. Then this RNA is converted by the reverse transcriptase into a DNA RNA hybrid. And the DNA RNA hybrid, the DNA molecule, the RNA will be used as a template to make a DNA strand, and this DNA strand will be used as a template to make another double strand. So now you have something like a cDNA. It's not cDNA, it's double-stranded DNA copy. And now this copy will be inserted into a new place within the gene. Okay. Some transposons, they are moving through a copy and paste mechanism. Some are through cut and paste. So some types of transposon, they cut themselves. They don't make a copy. They are cut from the genome and they move into another location within the genome. Okay? The transposons that move by the cut and paste mechanism, okay? And the copy and paste mechanism requires the enzyme transposase. Transposase. So a transposase is an enzyme that is required to insert a copy or a transposon into a new location within the gene. Retrotransposons always move by a copy and paste mechanism. So always a copy is left of the transposon in the original site. Okay? This retrotransposon, by the way, encodes. So within this DNA element, there's a gene that will encode the enzyme reverse transcriptase that is necessary to convert the transcript RNA of the retrotransposon into a double-stranded DNA molecule that could be inserted into another location within the genome. Okay? So I will stop here. Inshallah, we'll continue next week.